Today we shall discuss about a very interesting concept that is all about cell cycle, also about mitosis and meiosis. So what is cell cycle? It is nothing but it is the life cycle of a cell. Okay, so which will involve steps that requires the cell to grow in size, prepare itself for the division, and also it includes the stage of division. So it is the life cycle of a cell. The steps which are required to grow, prepare and to divide are all included in this cell cycle. So when we talk about cell cycle, there are two phases in cell cycle. One is the interphase and the other one is called the M phase. One is called the interphase, other one is called the M phase. What is this interphase is the time gap between one cell division to other cell division is what is the interphase stage. So this is the stage. Imagine that there is a cell. When it has to divide, first it will be in interphase stage. So during this stage, it is actually a resting stage or a preparatory stage, wherein the cell will prepare itself for the upcoming process of division. So this interphase actually has got three phases, namely the G1 phase, S phase and G2 phase. The G1 phase stands for the gap 1 phase, wherein the cell will grow in size, it will become bigger enough and it will develop all the proteins and other things that are essential for division during this particular gap period. Followed by this is the S phase or the synthetic phase. This is the phase wherein actually the DNA will duplicate, the DNA will double, so that when the division happens it can distribute the DNA to the daughter cells. The next we have is the G2 phase, which is the gap 2 phase. So this is the period during which the cell organelles, that is, let it be the mitochondria or the Golgi body, these sort of cell organelles which are present will divide, and the cell will check for any other mistakes that are there, and then it will enter into the next stage of division. So during G2 phase, the cell organelles divide and the cell will check for the mistakes and it will get ready for the process of division. The next coming to the next phase, which is the M phase. So once the cell is all ready, it has grown enough by taking in all the nutrients, and then it has well prepared by doubling its DNA, everything is ready now. Now it will enter into this M phase, which is usually called the mitotic phase, because an individual body is all made up of somatic cells. When the body cells of an individual has to be formed, when the growth has to happen, the most common process is mitosis. So M phase, you will write it as mitosis and cytokinesis. Actually in these processes of mitosis or meiosis, first the nuclear division will take place, which is also called the karyokinesis. Nucleus is nothing but it is also called the karyon. So karyokinesis is actually indicated in these processes, which will finally end up with cytokinesis. Cytokinesis is a process wherein the cytoplasm will divide and we have the formation of the daughter cells, which will be the final stage. So during mitosis, actually karyokinesis, that is division of the nuclear material, will take place. So let us first understand about mitosis. So simply to understand mitosis, if there is a 2n cell, you get two daughter cells which are also of 2n ploidy, diploid. So if there is a diploid parent cell, you get two diploid daughter cells in the case of mitosis. Suppose we humans have cells which will have 46 chromosomes. So once you are a zygote with 46 chromosomes, if this zygote cell has to divide and produce two more daughter cells by mitosis, they will also contain 46 chromosomes each. So that is nothing but the parent cell has got a particular ploidy, it will give rise to daughter cells with the same ploidy, whatever it may be. That will happen in the case of mitosis. So let us understand the process of mitosis through these diagrams. So this is the parent cell. Here you can see the parent cell. This is the chromatin material. That is, the chromosomal material will be present in the form of thin thread-like structures initially, which will be called chromatin material, and this is the nucleolus that is present. So this parent cell, after interphase, when it enters the M phase, what will happen? The first stage is called prophase. In prophase, what will happen? You see the chromatin material is all just thread-like like this, wound off. Here actually in the prophase, the chromosomes become thick, coiled or condensed they become, and they will split into structures which are called chromatids. See, a chromosome, when it is, when you draw it in the form of an X-shaped structure, then these two structures, which are there, these are called the chromatids. They are called the sister chromatids. So a chromosome will be made up of two sister chromatids. So during prophase, what is happening? Condensation of the chromosomes and also splitting of the chromosome into two sister chromatids will take place. So like that we will represent the chromosomes as chromatids like this. As you can see, the arms, there are chromatids, and then the centrioles will move towards opposite poles from where the spindle fibers will develop, and there is disappearance of the nuclear membrane. So these are the main events which will take place during prophase: disappearance of the nuclear membrane, condensation of the chromosomes, formation of the sister chromatids, movement of the centrioles towards the opposite poles from where the spindle fibers will start. So this is what is called prophase. Followed by prophase, we have the next phase, which is the metaphase. In metaphase, what will happen? The chromosomes which are there will come and get arranged in the central region, which is called the equatorial plate region, through which actually the spindle fibers will be moving through the centromere portion of the chromosomes. So like this they get aligned near the equatorial plate. An array you can see of the chromosomes near the equatorial plate. Usually if you have to see the chromosomes in a perfect form, metaphase stage is more suitable. They'll take even photographs of the chromosomes during the metaphase condition to find out any genetic anomalies. Usually the metaphase stage is preferred. So followed by metaphase, we have the phase which is called the anaphase. During anaphase, as you can see, the chromosomes, which are egg-shaped like this, they are made up of two chromatids. Each chromosome is made up of two chromatids, as we have seen. So these chromatids, with the help of the spindle fibers, the centromere portion will split and the chromatids will start moving towards the opposite poles. So each chromatid, so this is one chromatid, the other one is there. Each chromatid will start moving towards the opposite poles. So that will happen during the anaphase. So you can see like an umbrella structure under the microscope. Actually, when you are seeing the cells, you will see an open umbrella like that from any direction you see. Like an umbrella, you can see the chromosomes, the chromatids actually moving towards the opposite poles like that. So that is the anaphase. Next we have is the telophase. In telophase, actually you can see two daughter cells, with the separate nucleus. The nuclear membrane will be reformed, and the chromosomes which are there, the chromatids which have moved towards the opposite poles, they have decondensed and again they have become fiber-like. Nucleolus reappears. So this phase is exactly reverse to that of the prophase. So in telophase, actually we have formation of two separate nuclei with nucleolus, chromatin material, everything will get established. Followed by this, cytokinesis will take place, wherein the cell will divide into two and we have formation of two daughter cells with the same ploidy, that is 2n ploidy. Suppose if the parent cell is with the 2n ploidy, that is if it is a diploid, we have two daughter cells which are also diploid type, So like this the mitosis will proceed. You have to just remember that whatever is the ploidy of the starting cell, let it be 23, let it be 16, let it be 46, whatever is the starting cell chromosomal number, the same will be there in the daughter cells. And how many daughter cells are getting produced in mitosis? Two daughter cells with the same number of chromosomes. So that is all about mitosis. So hope you understood. So let us move on and find out about the next type of process, which is meiosis. So talking about meiosis, actually it takes place in the reproductive cells. That is, your body is all formed by mitosis completely. Even the reproductive organs will have all the cells which will have 46 chromosomes. But once a person enters into puberty stage, these reproductive cells which are there, they have to produce the gametes. Menstruation process starts in female, they'll produce the ova, and also the sperm production will start in males. So once that stage has reached in the individuals, gamete production has to be started. At that time, in the reproductive organs, the cells which are called meiocytes, in them meiosis will take place. So meiosis, actually it's a little bit complicated process which includes meiosis 1 and meiosis 2 stages. The meiosis 2 stage is exactly like mitosis. There is no difference at all. Whereas meiosis 1 we need to understand little bit. If you have to understand what is meiosis, in meiosis the parent cell, suppose if it is diploid, it gives rise to 4 daughter cells which are haploid. 4 daughter cells which are haploid. So haploid cells are very much essential for reproduction. The gametes have to be haploid for reproduction. That's why, because the ploidy is getting reduced here, meiotic division is also called as reductional division, whereas mitotic division is called as the equational division, whereas this is called as the reductional division. From a diploid parent cell, we have 4 haploid cells that you have to remember. So here, because the meiosis 2 is same as mitosis, I'll be focusing on the meiosis 1 process. Okay, just a brief understanding about the process. So in meiosis 1 also there is prophase 1, metaphase 1, anaphase 1, telophase 1. It will be called 1 and 2 there. So what will happen actually during the meiosis 1 process is, first in prophase. So prophase is little complicated. Here there are 5 stages: leptotene, zygotene, pachytene, diplotene, diakinesis are the stages. As you can see, okay, in the prophase 1, the chromosomes have condensed, okay, like that, and the homologous chromosomes will come together. Okay, that is, if there are 46 chromosomes in the case of humans, one will be from the father, one will be from the mother, the first one. Okay, the first chromosome from either of the parents will come, and that will be coming close together, which is called pairing. So that pairing will take place with the help of a complex called a synaptonymal complex. Okay, the synaptonymal complex will help in pairing. So first, what is happening in leptotene? There is condensation and coiling of the chromosomes with the formation of the sister chromatids. Next, in the zygotene stage, there is pairing. They come close together. Okay, the first chromosomes two will come together, the second chromosome from the mother and from the father, the paternal and maternal origin, they say, they come together. So like that, pairing will take place in the zygotene stage, and in the pachytene stage, what is happening? Crossing over. So that's why meiosis can lead to variations among the newly born individuals, because when the chromosomes have come close to each other, some of the arms will exchange like this. Some material of one of the chromosome will go to another, and this material will come to the other chromosome. So like that, exchange will take place. So what is happening? So here, pachytene stage, there will be crossing over between the homologous chromosomes. Crossing over, as you can see, they have crossed on each other. So like this. So in diplotene stage, there is a clear crossing over and appearance of an X-shaped structure like this, which is called the chiasma. It is called the chiasma. So okay, you find the chiasma in the case of diplotene stage. In diakinesis, what will happen? Okay, the chromosomes, they tend to move far away from each other, and there will be terminalization of this chiasma. Chiasma will be terminalized like this. Okay, so terminal movement. Imagine that two ropes have wound around each other. So when they have wound around, it is called crossing over. When you are pulling it apart in diplotene stage, okay, the knotted portion will be moving far away when you want to separate it from the middle. So like that, the chiasma, okay, undergoes terminalization in the case of diakinesis. So in prophase, what is happening? The main thing is, okay, there is pairing of the homologous chromosomes, there is crossing over, because of which some segments of one chromatid, okay, will get exchanged with the other chromosome. Okay, next we have is metaphase. In metaphase, there will be alignment of okay, these tetrads, that is the chromosomes which are attached to each other like that, they will be arranged along the equatorial plate like this. And then in anaphase 1, there will be separation of the chromosomes towards the opposite poles. Okay, so the chromosomes will move towards opposite poles like this. So because of this, what will happen? Okay, the chromosomes have moved towards opposite poles, because of which what will happen? The reduction of the chromosomal number will take place. That is, from a 2n cell, okay, you are getting 2 cells, okay, which are n and n ploidy. Okay, which are n and n ploidy. That is, half the chromosomes are moving towards one end, half the chromosomes move towards the other end, and there will be formation of the nucleolus, nuclear membrane, etc. in the telophase 1. And there will be cytokinesis. So what has happened? From one cell, okay, we are getting 2 cells which are having just half the number of chromosomes. And this is the significance of meiosis, okay, wherein the reduction has to take place for the formation of the gametes. Then next, other steps will be same, like, so imagine that these 2 cells have entered the mitosis process, and all the steps are getting repeated. So finally, after the end of the meiosis 2, here, these 2 cells will enter into meiosis 2, which is same like mitosis, and they will result in the formation of, these are, the parent cell are having n as their ploidy. So from each of the parent cell, you get 2 daughter cells with n as the ploidy. Same number of chromosomes will be there in the daughter cells. So from 2 cells which are having n as their ploidy, we are getting 2 more cells from each of the cell. So finally, by the end of meiosis, we have 4 cells. Out of 2 cells, we have 4 cells. Okay, 2 cells of meiosis 1 give rise to 4 cells, okay, of the same ploidy by the end of meiosis 2. So like this, we have the mitosis and meiosis, which will be proceeding like this. Meiosis as such, they'll not ask much about that, but mitosis, if you are confident with this is more than enough. For your understanding, I have given a brief explanation even about the meiosis. Any doubts if you have, you can post in the comment section so that we can discuss about all your doubts in a clear way. Hope you followed.